Ladies and gentlemen, welcome to the Select Bio webinar sponsored by Spectrodyne this morning. And for those of you in Europe, good afternoon, good evening. We're delighted to welcome an excellent set of presentations that will be delivered as part of this webinar today. The first presentation will be by the CEO of Spectrodyne, Jean-Luc Fréquin. And then the second presentation after Jean-Luc's talk will be by Zoltán Varga from Budapest. So without any further ado, ladies and gentlemen, let me welcome Jean-Luc to deliver the opening presentation of today's webinar. Welcome, Jean-Luc.
Great, thanks, Anil, and thanks everybody for joining us today. As we mentioned already, so I'll give a brief overview of how to use Spectrodyne's technology, the NCS1, to quantify viruses quickly. And then I'll hand it over to Dr. Varga, who will present some exciting results of using it to measure SARS-CoV-2 virions. First, let's review, why we're all here. So why is virus concentration so critical? And the answer is basically because it's a critical experimental variable, right? And the better we can control those, then the tighter our science will be. This comes up in two main applications with viruses. First, when we're using viruses as a therapeutic, so for example, gene therapy or vaccine delivery, right? In this case, the concentration of the virus is really the dose of the product. And so it's critical to control it carefully. Leads to safer, more effective products and better product development. Second case is when we're using viruses as an assay readout. So that would be, for example, in diagnostic testing. And here, the concentration of the virus is the test result, right? So the better we can measure that, the better your assay sensitivity and specificity can be. Bottom line being that the better we can measure concentration of the virus, then the tighter our science will be and the faster you can move in your research. So the ways people are doing it, currently that include live biological titer. This is a gold standard, but if you've ever tried to do it before, you know how painful it can be. Results take, at best hours and as long as days. It's error prone, takes a lot of replicates, and you have to do cell culture, which is very time consuming and costly. Obviously, there are molecular methods like PCR and protein quantification tools. These are slow also, and they can be really complex, especially if you have to develop your own assay for a new virus application. And they're still slow. Recently, there are more particle analysis methods. So for example, nanoparticle tracking analysis, or even dynamic light scattering some people use. But these are really challenged by the application because viruses by nature are weakly scattering biological nanoparticles. And so that makes it very hard for these light scattering tools to do it. Plus, they live in complex samples like cell culture media or serum that very quickly overwhelm the optical methods, so it's not really practical. Spectrodyne's technology is a cutting-edge microfluidics-based technology that delivers accurate virus titer from 50 nanometers diameter and up. It uses electrical detection, so not optics, to measure each particle as they are detected, and delivers results in minutes. So we can measure concentration of the virus in minutes. It's easy to adopt. Because we're using microfluidics, only a very small volume of the samples required to measure, and the cartridges are single use, so there's no cross-contamination. It works using microfluidic resistive pulse sensing, which is essentially a modern version of the Coulter principle, which has been the gold standard for measuring large particles in cells for decades, really. The way it works is the whole sample is flowed through a bottleneck in the fluid flow. And every time a particle goes through this constriction one by one, it generates an electrical signal. We measure basically two things about the electrical signal. One is how big it is. So how big the spike is tells us how big the particle is. And how quickly the particle goes through the constriction tells us the flow rate of sample through the constriction. So that allows us to calculate concentration. This is a longer measurement, real data showing measurement of many particles, and you can see just by the bands of signal amplitudes that are being detected here that there are 4 different populations of particle sizes in the sample. And just counting them up, looking at the histogram, we can get that information. But we can go a step further because we know the volume of sample that's been measured, and so we can convert these counts to absolute concentration. And this is the final product of what our technology does. Essentially, it's a plot showing concentration on the y-axis versus particle diameter on the x-axis. And we can be quantitative over any size range here. This example was just a mixture of polystyrene particles, 50, 90, 120, and 150 nanometer in diameter. So it's a very easy sample. But this combination of accurate concentration and size, together with the ease of use of the microfluidic chips and the instrument, just not available with any other method currently. Let me show you a quick measurement example of human adenovirus 5. And I wanted to start by showing you what the raw data looks like. So that's what this is. You don't have to look at this when you're doing the measurement, but you can. And what we see is a smooth baseline. And every time a virion goes through the sensing constriction, we get an upward spike that's been highlighted with a red dot here. And so essentially what we're doing here is seeing the viruses as they're being detected one by one. This happens very, very quickly, obviously, so you can tell by the x-axis time scale here. So when we do the counts of all these particles and make the size distribution, this is what we get. This is showing 3 replicates, technical replicates of the measurements of the same sample. There's a very clear identifiable population for the virus at around 90 nanometers, which is where we expect for this virus. And as I mentioned, we can be quantitative over any size range you want here. So maybe you want to measure the virus concentration, or maybe you want to measure off-peak concentration to quantify impurities, things like that. For the virus peak itself, we just measure the concentration over that size range, and you can see we get excellent repeatability. Remember, this is absolute concentration of virus particles, about 5 minutes per sample without any tissue culture. So this is a pretty easy 15 minutes or so measurement to do these replicates. How can we use this technology? So let me show a couple of examples of that. First, in developing virus purification protocols. So this example is showing measurements of 3 different samples. First is the crude virus preparation, which is shown in the dark blue here at the bottom. It's got a broad distribution of particle sizes. There are contaminants from the cell culture process, right? And there's no discernible populations in there. But as the sample is purified, a peak due to the virus emerges right where it's expected in size. And this information, because we can measure the concentration of virus, right, can be used directly to improve the purification process. And this customer found this information extremely valuable because it validated their purification number 2 process. Next example I want to show you is a little bit more of an academic example, a researchy example. This shows measurements of 2 flu B variants. One is a wild type virus, and that's in blue. It shows a nice clear peak around 100 nanometers where it's expected, and those are the virions being detected. Compared to, in green, what we're showing is the mutant virus. And in this case, the mutant clearly is affecting the ability of the virus to create capsids. And so that's valuable information about how the molecular changes made to the virus affect its physical structure. Finally, I want to highlight this example because this is something that our technology can do like no other, and that is because the measurements are so fast, it can allow real-time process optimization. So, for example, this measurement is showing crude myxoma virus prep. The particle size distribution includes a lot of, background particles, right? A broad distribution of background particles. And the myxoma virus is present here as a peak in this distribution as expected around 260 nanometers diameter, so effective spherical diameter. So what's useful here is that we can quantify this virus in the crude prep and monitor that concentration as a function of time to optimize the harvest time. So when we zoom in on this virus peak in the distribution, we can measure just the virus particles in here. Over this size range, get about 2 to 8 particles per mil. Because it just takes a few minutes, we can keep sampling this until the concentration is optimized for the production efficiency. Let me show you just 2 examples that demonstrate the performance of the NCS1. First, we did a serial dilution of a retroviral drug formulation, and you can see we get a nice linear response, excellent linear response in concentration as a function of dilution factor. And finally, let me just show you the dynamic range of the technology. So this is a measurement of a single sample spanning from 65 nanometers diameter all the way up to 6 microns particle diameter. And you can see that in this single sample, the concentration varies as a function of diameter over 9 orders of magnitude. There's just no other technology that can make such broad range measurement currently available. So hope you've learned something about our technology today. It's a small benchtop instrument, uses microfluidics to deliver accurate viral titer in a few minutes. That allows you to do better science, basically, and that's what we're here for. I showed you just a few examples. Our customers use the NCS1 to quantify many different samples, not just viruses though, too, EVs, protein aggregates, and other drug delivery vehicles. If you still don't see your sample on this list, reach out to us. We love to demonstrate the technology, and we would love to receive a sample for you and do a free measurement to show you the power of this one. So thank you very much for your time. At this point, I will hand it over to Zoltan and from Research Center for Natural Sciences in Hungary, and he can show his examples of using the NCS1 to detect SARS-CoV-2. It's very exciting. Thank you.
Thank you, Jean-Luc. So now we will make Zoltan the presenter, ladies and gentlemen. Also, if you have questions, you're welcome to type them in the question box in the control panel in the upper right-hand corner of your screen. Alternatively, at the end, you can raise your hand electronically and we'll unmute you and you can ask the question in real time. And we'll do the Q&A. Myself and Lou will be doing the Q&A at the end of Zoltan's talk for both Jean-Luc and for Zoltan. Welcome, Zoltan. Thank you.
Hi, thank you, and welcome everyone. So in this short presentation, I would like to talk about the single particle detection of the SARS-CoV-2 variants by microfluidic resistive pulse sensing. Let me start with the size ranges of viruses. So it covers a wide size range from 10 nanometer up to several hundred nanometers. The most common viruses like influenza, adenovirus, HIV, and also the coronavirus falls in the 100 nanometer size range. For the novel SARS-CoV-2, it is reported that the size of the virus is in the range of 100 nanometer to 120 nanometer. These observations are based on mostly on transmission electron microscopy and cryo-electron microscopy. It should be mentioned that these size estimates are based on counting single particles, several hundred single particles. In this work, our goal was to use an orthogonal technique, I mean orthogonal to the microscopic techniques, to measure quantitatively the size distribution of the novel coronavirus. So for this, we use microfluidic resistive pulse sensing, as it was introduced by Jean-Luc. This is a non-optical method which can detect particles from 50 nanometer up to 10 microns. As you could see, this is a single particle detection method, but due to the high particle detection rate, which can be several thousand particles per second, this method can give quantitative results on the size distribution. So in this technology, there are different cartridges for the different size ranges. In these particular experiments, we used the TS400 cartridges with NCS1. So this cartridge covers the size range from 65 nanometer up to 400 nanometer. And the maximal concentration which can be measured here is 10 to the 11 particles per milliliter. So the sample preparation, so viral cells were grown and they were infected with the coronavirus with the multiplicity of infection of 0.1. The control sample was the cell culture supernatant without infection. The sample processing was kept as simple as possible. So in the first step, the cell and debris was removed by centrifugation at 800 G for 10 minutes. Then the cell-free supernatant was further filtrated with a syringe filter of with a pore size of 0.45 micron. And in the final step, we concentrated the sample by approximately tenfold with tangential flow filtration. Here on the right, you can see the control cell, the microscopic image of the control cell layer, and the Vero E6 cells infected with the coronavirus and incubated for 84 hours. All these experiments were performed in the BSL-4 virus lab at the University of Pécs. Actually, the cell work and the filling of the cartridges was performed in the BSL-4 lab. Then the cartridges were placed in a closed container and transferred to the BSL-3 lab, where the instrument was installed and the measurement took place there. Let me start with the size distribution of the cell-free supernatant without infection. Here, the red symbol shows the size distribution of the supernatant after the background correction. As you can see, this is a monotonic decaying size distribution, which is characteristic for the size distribution of extracellular vesicles produced by either by the Vero A6 cells and also the serum, which was in the medium. This is a linear plot. If I change to a double logarithmic plot, one can observe that the size distribution follows a straight line, which means that this size distribution, so this is the background from the extracellular vesicles, this size distribution follows a power law function. When we look at the raw data from the cell-free supernatant after infection, we can already see on the raw data that a huge population appears in the small size range. If we zoom in to one single detection, we can observe that this population corresponds to sizes a little bit below 100 nanometers. If we construct the size distribution from these raw data, we can obtain the distribution shown with the green symbols. In contrast to the cell-free supernatant without infection, here we see a clearly resolvable peak below 100 nanometer, which can be attributed to the virions. It is interesting to notice that in the very small size range, the non-infected cells produce more small extracellular vesicles than the infected ones. If we analyze the size distribution of the SARS-CoV-2 variants, we can observe that so this distribution was constructed by counting more than 45,000 particles. That was a single 5-minute experiment. And here the main diameter was found at 84 nanometer with a standard deviation of 10 nanometers. And here the concentration of the virions was 8 times 10 to the 9 particles per milliliter. So this is the concentration after concentrating the cell-free supernatant, the infected cell-free supernatant with tangential flow filtration. Okay, the first question what can be asked here is why we measure a peak of the size distribution below 100 nanometers, since most of the papers reports 120 nanometers. First, we should consider that the MRPS measures the effective diameter corresponding to the excluded volume. If we look at the latest cryo-electron tomography results from SARS-CoV-2, we can observe that the envelope diameter, which is defined by the diameter of the phospholipid bilayer, falls below 100 nanometer. It has a peak at 80 nanometers, and the size of the spike proteins extends up to 20-25 nanometer. So if we consider the volume of the spike protein and the average number of spike protein per virion, which is around 30 proteins per virion, we can calculate the effective volume of the complete virion, which is the volume defined by the envelope plus the total volume of the spike proteins. And from this effective volume, we can calculate back an effective diameter, which would corresponding to the diameter which is measured by MRPS. If we take 80 nanometer as an envelope diameter and all these numbers which I just mentioned, then we get an effective diameter of 81.5 nanometers. So this means MRPS practically measures the envelope diameter of SARS-CoV-2, which is slightly above 80 nanometers. So this is the take-home message of this talk, that MRPS can be used to quantitatively measure SARS-CoV-2 in liquids, and the diameter what we obtained by MRPS regarding SARS-CoV-2 corresponds to the envelope diameter. With this, I would like to thank my collaborators at the University of Pécs in the virological research group, and also my colleagues here at the Research Center for Natural Sciences. I'm happy to take any questions, and thank you.
Thanks very much to both Zoltan as well as Jean-Luc for their excellent talks. Ladies and gentlemen, the webinar is now open for questions, Q&A. You can either type your question into the question box on the upper right-hand corner of your screen in the control panel. Alternatively, you can raise your hand and we can unmute you. There's a number of questions which my colleague Lou Brown and I will take in turn. So the first question I will read out. It says, and this is for both Jean-Luc as well as Zoltan, how can you differentiate between viral particles and particles of similar size, such as extracellular vesicles, exosomes, using this technology? So this question is for both of you. So if you would kindly address. Thank you.
Sure, that's a good question, and fundamentally, this technology does not distinguish between a virus particle and an EV, right? But I think what Zoltan has showed us is that it's pretty obvious to see them. When you look at the particle size distribution and you do your experiment with an appropriate control, then you can see them as they, form a clear population in the particle size distribution. Same with the data I showed you too. So it does depend on the purity of the sample, but Zoltan did very little to purify these samples before measurement. So I think this shows the applicability of that. There's also, I'll just add quickly too, there's the ability to subtract a blank background also. So the way Zoltan did it was he measured the empty, the uninfected background, and there are tools in the software for subtracting a background blank, essentially, to give you the difference. Did you want to add anything to that, Zoltan?
No, I just agree totally with your answer. So in fact, the same particles with the same size cannot be distinguished, but the analysis of the size distribution clearly shows if it is an extracellular vesicle sample, because then we measure a power law type of distribution, and for viral particles we see always a peak corresponding to the size of the virions.
Thank you, gentlemen. Lou, would you like to take the next one, please? Thank you.
Sure. Let's see. Comment was beautiful data. What is the flow rate divided by sample volume, or flow rate or sample volume?
I can't answer offhand, obviously, but typically the flow rate is in the range of, well, for the cartridges you were using, Zoltan, do you know offhand what the volume measured, the total sample volume was?
By heart, I don't know, but it is in the, I don't know, femtoliters, right?
Yeah, small amount, right? Yep.
A very small amount. And one run.
Yeah, go ahead.
Yeah. So one measurement is 60 seconds, and in 60 seconds we measure I don't know, thousands of particles.
Right.
Great. So there's many more questions. Let me read the next one. How would you use an appropriate control from a human biological matrix? Would it be plasma or serum? So either of you gentlemen to address.
Go ahead, Zoltan. It's a science question.
Okay, so maybe I don't understand properly this question. So the question concerns what is the difference for plasma and serum, or can we measure plasma and serum? Well, for sure, both we can measure. We also measure the size distributions in plasma and serum. Of course, here it depends on the sample processing, but in case of plasma, we will measure of course, extracellular vesicles and lipoprotein particles, VLDL particles, and chylomicrons. In case of serum, we did an experiment. So we compared the concentrations, the particle concentrations in plasma and serum, and we observed higher concentrations for serum, and we attributed that to the appearance of platelet-derived extracellular vesicles, because during blood clotting, platelets get activated and they produce a lot of particles. And on this size range, both shows a typical power law type distribution.
Thank you. Lou, for the next question, please.
Sure. Let's see. How do you decide where to gate? So talking about filtering, I think.
Shall I
Sorry.
start? Okay. So yeah, in practice, we always look at the background noise. So we look at the plot. I didn't show the scatter plot, which plots the transition time and the diameter of the diameter versus transition time, and there we can discriminate real particles from the background.
Yeah, I'll just add that the software does automatic gating of the electrical noise that works fine for most samples, and in some cases, you can customize them to meet your application.
Okay. There's an interesting question. How does your particle count data correlate to infectious titer or genomic titer for the sample? So your data versus infectious units, IFUs.
Zoltan, do you have an answer for your measurements?
Yes, I can just say that we did qPCR check, and now droplet digital PCR measurements are carried out. And the first preliminary experiment shows that in this special case, it kind of correlates. So with PCR, we get the same order of magnitude to what we measure with MRPS for SARS-CoV-2.
Right. We've done other experiments with phage particles also that show tracking between, correlation between the PCR measurements and the actual virion counts. It's my understanding, I'm not a cell biologist, but it's my understanding that there's a lot of different variables at play in converting between an infectious unit and the particles, but that it can be essentially calibrated based on the system. So just as you don't necessarily use copy number as being equivalent to virus number or even infectious units, it can be the same thing for the MRPS, the virion count.
Okay. Lou, for the next question, please.
Sure. Is there a reason you didn't include standard beads in your measurements of the SARS-CoV-2 variants?
Yes, in fact, we did measure 90 nanometer polystyrene beads and calibrated the cartridges. By the way, those are also factory calibrated. We just checked the calibration for the certain cartridge that we use for the virus measurements.
So there's another question. Molarity-wise, what was the concentration of SARS-CoV-2 virus in Zoltan's presentation, or was it a random concentration directly taken from a patient?
This sample was from a cell culture, and there the cell number was approximately 5 million per flask, and the concentration that we obtained corresponds to the concentration of the sample which was concentrated with tangential flow filtration. So it was not a patient sample.
Okay, thank you. Lou, for the next question, please.
Yeah, so this is a little bit of a related question, or it's a comment, which maybe Zoltan, you go a little more further on. Just EVs are also different in healthy versus sick people, so you can't be sure using a healthy uninfected sample as a control. It sounds to me like you're saying you didn't use a, I don't know, you interpret that. I'm not sure there.
Okay, this question corresponds or relates to the question of what could be the good negative control in these experiments. Again, I can repeat that if you look at the size distribution, we see power law, so monotonic decaying size distribution for the non-infected cell-free supernatant. And in case of the virus-infected cells, in the supernatant, we see a peak, a clearly resolvable peak. So it's obvious that this peak comes from the viral particles. Of course, since the viral infections already has an effect on the production of EVs by the cells, so a direct subtraction will not work here. But of course, one can think about how to make these measurements more precise. We also started to work on separating EVs from virus particles, but since they are the same in size, we need to use very elaborated techniques like density gradient, and which is very hard to carry out in the virology lab. So, but my answer is that already from looking at the slope and the shape of the size distribution, one can distinguish what is coming from extracellular vesicles and what is coming from the virions.
Thank you. There's another question. Again, I think this goes to Zoltan. Is there a reason you didn't include standard beads in your measurements of the SARS-CoV-2 variants?
Okay, it's almost like the same question which was before. So I didn't show the measurement of the standard beats, but we did measure the standard beats. If the question relates to why didn't we use a spike in experiment, and the answer is that I have a calibrated reference standard, which is polystyrene with 90 nanometer in diameter. So if I spike in the virus sample with this, so there is an overlapping size. So for a spiking experiment, one should use a 200 nanometer or something below that. And simply, we had one day to work in the BS4 lab. There was no time for doing these experiments. So these were just preliminary experiments, but further research is planned.
Thank you. Lou, for the next question, please.
Yeah, sure. Good question here. How do you avoid clogging of the pore?
Good question. So the pore is not isolated. So these are in an engineered microfluidic cartridge, and upstream from the pore is a built-in filter. Essentially, it's just a series of small channels that generally excludes particles that would be, too big to fit through the pore. So this allows faster running. Of course, not perfect, but as Zoltan's data shows, it's easy to measure many thousands of particles in a complex sample in the lifetime of a cartridge.
So, ladies and gentlemen, if you have any further questions, you're welcome to type them in to the question box in the control panel. There's a few questions that the team will take offline since they're very specific to the very to the user's experiment. But at this point, I'd like to ask both Jean-Luc and Zoltan to make any final comments regarding, for instance, Jean-Luc on the platform, as well as Zoltan on the usage, and some closing thoughts, because we have time, and I think it's been a very engaging webinar with lots of questions. So Jean-Luc, if you'd like to go first, please.
Sure. Yep. Thanks. So I just encourage anyone that's interested to learn more about the technology to reach out to us, visit our website, and to contact Zoltan to learn more about his cool science that he's doing with it. Thanks very much, Zoltan, for joining today and for presenting this research. I'm looking forward to seeing more in the future too.
Yeah, I also thank you for the opportunity. Yes, I can just add that, of course, we are not only measuring virus particles with NCS1, but we also measured already nanoparticles, artificial silicon nanoparticles, for example, or liposomes. And in all cases, the NCS1 was found very easy to use and produced nice results. Of course, there are technical parameters and other stuff that needs to be optimized for each type of particles. But in overall, this is a novel technique, orthogonal technique to the widespread methods like NTA and ELS. Thank you.
So we'd like to thank both our speakers for a very engaging webinar. Both Jean-Luc and Zoltan will be speaking later in February at the Select Bio EV conference, where Jean-Luc will provide a deep dive on the Spectrodyne platform for extracellular vesicles EVs, as well as virus work that they're doing. And Zoltan will again be providing another presentation. So those of you that are interested, you're welcome to check out the website. We'd like to thank Spectrodyne for once again a great talk. Thank you, ladies and gentlemen, for your engagement and lots of questions. This is great. Please keep them coming. And I'd like to turn it over to Lou for any final comments before we close the webinar. So Lou, I leave over to you. Thank you.
I really don't have any final comments beyond to thank both the speakers and particularly Zoltan. It's really interesting data. I actually had one question myself, which is how is the 100 nanometer diameter from the EM images? How do they come up with it? Does that include, do they measure across the whole particle, including the spike proteins, or how did they come up with that?
Those are just size estimates and looking at the maximum hundreds of particles. And actually on the cryo EM paper, they measured almost 3,000 particles. The first estimates coming from transmission electron microscopy, I think that correspond to the overall size, so including the spikes. And that, so if you look at the cryo-EM data, if we just add the 80 nanometer envelope diameter plus 2 times the 20 nanometer, which corresponds to the height of the spike, then we get 120. So I think that's the answer to this question.
Yeah, great, thank you. That's very helpful. That's really all I had. Thank you for hosting, and thank you everybody for attending.
Thank you, everybody. Any further questions, please email to us or email to Spectrodyne directly, and Jean-Luc and Lou will be happy to address them. So hope everybody has a great day. Thank you to all the speakers for excellent presentations, and goodbye. Thank you.
Thank you.
Thank you. Goodbye.